Yes. Yeah. Look, pretty much just walk straight out of those doors. Past one. Just like one block past one. Yale.
You all set?
Yeah.
Do you have the mic on, by the way?
Yeah, dude.
No way. All right. Welcome, everyone. Thank you for coming. I'm Ian Winchip. I am Sam. So we're here for Sam Joshua Basco-Jusidas's PhD defense talk. Now, looking around the room, I think most of you know Sam. He's been here all the way since 2011, and he's been working in that time with me and co-supervised by Catherine Todd. He arrived from Taiwan with a master's in molecular biology and then started doing something completely new. One of them was almost dying in the first winter in 2011 and 12. I've never seen anybody wearing so many clothes and still being so cold, but he made it. And later that year, he started what was initially going to be a research rotation project in Catherine's lab. And it was a really basic question that I had because I was interested in the plasticity of the spinal cord. And so I thought, well, maybe we had some evidence that inflammation and plasticity in the cord were correlated after stroke. And we thought, okay, well, maybe the microglia are a little different. We thought it was going to be a few months of work, and 5 years later, Sam is going to tell about his work on functional characterization of spinal cord microglia.
Thank you. Good afternoon, all, and I really much appreciate you showing up here on a Thursday afternoon and for my talk. So, for that sense, I'll try to make it as interesting as possible, and also, as holistic as possible, so you could get the story of what I'm trying to say or express. So that is my thesis title, and I'm going to present on my thesis. And this is my PhD public seminar. So officially, I thank you once again. My research topic is functional characterization of spinal cord microglia. So let's jump into it. I'm going to start somewhere outside microglia, inflammation, and there's a reason why I'm talking about inflammation. Inflammation is a complex biological response. So what it is you have, say, a pathogen, an injury to the brain, the immune cells come in, and they are needed to patch up things. They fend off the pathogen or phagocytosis, they heal the brain or the central nervous system, and again, they phagocytose and provide an environment where the cells can recover. So it is essential for healing process. So that's one side of inflammation. But there's also other side to inflammation. It's not a fixed, process. It's a gradient. So on one side, it can go progressively and become detrimental to the system. On the other hand, it can be progressively healing. So that's kind of like inflammation. Now, inflammation itself has a huge role in central nervous system disorders, like diseases and disorders. This is just a brief list of the global burden of inflammation, and there is a component of inflammation in all of these diseases. This is not a comprehensive list; it's just a few diseases. So, microglia are the primary immune cells of the central nervous system, which means they are the initiators of inflammation in central nervous system. They are the resident immune cells, like I said. They were discovered by Ortega, and he found that, or he postulated that, these cells are constantly surviving the microenvironment. The green blobs are microglia, and the stuff that's moving around is their processes. So they're constantly surveilling the environment. When there is an injury, like here, there's a focal injury, they immediately send their processes and then shield that injury site. And that's the beginning of inflammation. They shield it, they initiate factors that would recruit more immune cells like microglia and peripheral immune cells. They come in, they fight phagocytose, and then they help in the recovery of that injury. So how does microglia do that, and what can they do? So, like I said previously, they have this state where they are constantly surveilling. We call them ramified microglia, and this state is considered non-active, and it could be due to survival signals that are present in the extracellular milieu. So, your central nervous cells, primarily neuron, send out signals to microglia saying everything is okay, we are calm. However, if there's an injury, what happens is the ramified microglia transform to activated states. There's several states, but for simplicity, we call them amoeboid and circular. The amoeboid are considered secretory, which means they release toxic and trophic factors, and the phagocytic microglia clean up. They eat a lot, and they are also capable of releasing factors. So that's a cycle. They initiate it, they continue on with that process. And how does this process start? So that's where these guys come. The factors that can initiate microglial activation can be categorized into two. First one is pathogen-associated molecular patterns, and from the name you can say it is associated with pathogens. Some of the pathogens that can mimic this are bacteria or virus. It could be an outer membrane of a bacterial cell wall, for example, like lipopolysaccharides. So these primarily activate microglia to toll-like receptors. We don't have to worry about these receptors right now, but as we go, we'll talk more about them. On the other hand, danger-associated molecular patterns are not foreign bodies, but they are locally generated. So these are factors that are released when a central nervous system, a cell, or it's dying. Its membrane explodes, it releases factors, and that tells them there's something happening, the system is compromised. Examples of these factors, danger-associated molecular patterns, are ATP, glutamate, cellular debris, and the list goes on. So these guys initiate inflammation or activate microglia. So more on that, so PAMPs and DAMPs can activate microglia, and that puts them in these states. There is a pro-inflammatory state, an anti-inflammatory state, a trophic state where they promote growth. And the balance of this is complicated. We don't quite understand that yet. But it goes back to this gradient that I was talking about, where, for example, if you take one pro-inflammatory factor like TNF, studies have shown that TNF is pro-inflammatory. What if we just block it? And will that make it a healing process? In stroke, just blocking TNF does not generate healing. Actually generates healing. Blocking generates healing, but if you remove TNF completely, which means there's no TNF in the system at all, it exacerbates the injury. So that's telling that inflammatory factor is needed, but it can work both ways. Another thing, what about we take microglia completely off the system? What if we don't need microglia? That also is problematic. If you remove microglia completely, it increases the injury. And if you introduce microglia early on into an injury, it increases recovery or healing. So, that's kind of telling. Early on, these cells are needed, and early inflammation or acute inflammation is needed. And acute pro-inflammatory factors might be actually providing for healing, versus chronic inflammation could be detrimental. And that's just one way of seeing it. There are several ways we can try to figure out what makes a microglia toxic or trophic. That's for another day. So with that, it's not just the microglia. If you look at the central nervous system itself, there are regions within the central nervous system that are vulnerable to injury. Again, one favorite example that Ian always talks to me about is hippocampal cells. They are much more prone to ischemic injury compared to other regions of CNS. So the order is, if there is a global ischemia, hippocampus is more injured, followed by cerebellum, followed by cortex. So that's vulnerability. So that's for an acute injury. How about neurodegenerative diseases like Parkinson's, Alzheimer's? Um, Studies have also shown that there is regional dependence. There are certain regions that are more prone for lesions. There are certain regions that are more prone for where Parkinsonian-like symptoms, evolve or grow. So that's telling that inside the brain itself, certain regions are more vulnerable for inflammation-mediated neurodegeneration. Similarly, certain regions in the brain are more prone for ischemic injury-mediated inflammation. So that kind of covers that. More studies from our lab actually show that there's also an age-dependent component where as you have aged microglia, they lose their effectiveness as inflammatory factors. So basically, there is all these factors that have been characterized, but all of them has been, or the studies have been done on brain microglia. But in the central nervous system, most injuries are not just related to brain. There is spinal cord. There is injuries that also occur in spinal cord. So can we relate what we studied in brain in spinal cord? So that's where I come in. I said, well, let's check it. Let's compare brain and spinal cord microglia and see if they are comparable. So for that, we had to, it's been this long because we had to establish protocols to get spinal cord microglia from neonates. We are one of the few labs that actually have successfully done that. There's very few studies for isolating spinal cord microglia. So it's a very, the idea is very simple. You isolate brain microglia and you also isolate spinal microglia. Here they are labeled with IBA1. Just remember this molecule, we'll be seeing it a lot. It's a marker for microglia. So we isolate these cells from brain and spinal cord, and then we activate them with, if you recall, Hans and Dans that I talked about before. And then we compare their activation profile. So the first question was, you know that a PAM, a pathogen-associated molecular pattern, will strongly induce inflammation in microglia. Then we want to test, okay, so let's use LPS because we know it's a bacterial toxin, it's a potent mimic of bacterial pathogens. Also, it induces pro-inflammatory molecule release. So it covers all the bases for us and also increases phagocytosis in microglia. So now, using this, if we compare, we can have a spectrum to compare against brain and spinal cord microglia, and that's what we did. So we applied LPS to brain and spinal cord microglia, and we measured their morphology, we measured pro-inflammatory and anti-inflammatory factors, and finally we also looked at phagocytic activity. So I know it's a busy slide, I'll kind of take you slow. So this is to kind of refresh you on the morphological states that I explained prior. So that's a ramified microglia where you can see all the processes are nice spread out. That's an amoeboid microglia and a circular microglia. So in this experiment, what we're trying to understand is if you activate brain and spinal cord microglia with LPS, are we seeing changes in these shapes or morphologies? And we did find some changes. In y-axis, we were looking at the shape parameter. So for ramified microglia, it's not significant, but there's a trend where there was increased presence of ramified microglia in the spinal cord and not in the brain. And after activation with LPS, compared to brain, there was still a trend for a higher presence. For amoeboid microglia, which if I remind you guys, if I can remind you, amoeboid microglia are considered secretory, and in the brain, LPS activation, when you compare LPS activation in brain compared to spinal cord, there was a decrease in that amoeboid form. And this is suggesting that, okay, there's a decrease in amoeboid form, so is there potential decrease in factors released by spinal cord microglia. For the spherical morphology, we did not find across group differences, but we did find that LPS increases this phenotype, which again fits with the idea that LPS is a strong activator. It will induce phagocyte-like phenotype. And so the summary of the slide is we found that there was a decrease in amoeboid morphology only in spinal cord when compared against brain and spinal cord microglia. So with that, we looked at the pro-inflammatory molecules. The first three of them, nitric oxide, trienophenol, and beta. Nitric oxide is it's a well-characterized pro-inflammatory molecule. It generates reactive oxygen species. and it is actually detrimental to the cell. Higher concentrations can destroy the cell. When we measure nitrites in the media, nitrites is a proxy for nitric oxide. We measured it in the media, we found that brain microglia had increase in nitric oxide release, and spinal microglia also had an increase compared to control, but that increase was significantly lesser than brain microglia. TNF is another pro-inflammatory molecule. It has multiple roles. One is downstream. It can induce the release of other pro-inflammatory factors on one side. On the other side, it can also induce apoptosis. So it kind of has dual roles. Similar to nitric oxide, we found that activation with LPS increased TNF in brain microglia. However, in the spinal cord was significantly lesser than the brain. IL-1 beta is also another pro-inflammatory factor. It has roles in neurotoxicity, and it had the same situation where activation with LPS increased IL-1 beta in brain, but we didn't see any effect of it in spinal cord, and it was also reduced. So this is for the pro-inflammatory factors. What about other factors? In my introduction, I introduced there is the anti-inflammatory factor. There is also the trophic factors, and IL-6 is commonly a pro-inflammatory molecule, but sometimes it can also prevent apoptosis. So it also has dual roles. When we activated brain microglia with LPS, we found that there was a trend towards increase in IL-6 in the spinal cord compared to brain. For IL-10, we did not see any meaningful differences. And also for BDNF, we did not see any meaningful differences. So the summary for this would be that there is a trend for an increase in IL-6, but other factors like anti-inflammatory and trophic factors are not much different in the brain or spinal cord microglia. So the overall idea is then how about functional differences? So phagocytosis assay is a very functional assay. And it tells if the cells are active. The way this experiment is done is again microglia labeled in red. We apply fluorescent beads. These beads are uptaken by microglia. So one would assume if microglia are activated, they will take up more beads, and if they take up more beads, that can be measured as fluorescence. So here again, we found that in control, that's normalized to control. We found that LPS induced an increase in bead uptake in brain microglia, but we did not see an increase in spinal cord microglia. So we started here. We wanted to check what happens if you mimic a pathogen like inflammation, brain or spinal cord microglia. So we activated them with LPS, and we found that there was decrease in amoeboid morphology. To time with that, also pro-inflammatory molecules secreted were reduced compared to brain microglia. The phagocytic activity was also reduced in compared to brain microglia was reduced. So this is telling that at least for a PAMP, the response profile of spinal cord microglia is attenuated compared to brain microglia. And so I would like to recall previously I said, we looked at PAMs, now let's also look at DAMs. And the reason we want to go back to these molecules is they are released in the CNS, they are more like a physiological activator. And so we picked ATP and glutamate because, at least in a stroke-like injury, these are molecules that are released immediately into the extracellular environment. And so we wanted to see that, do we see similar differences in brain and spinal cord microglia, like so what we saw for LPS. We also postulated, based on the LPS study, that DAMs would have a reduced inflammatory profile on spinal cord microglia. So that was our question and our hypothesis, and the experimental paradigm is very similar. We have brain microglia, isolated spinal cord microglia, activate them with ATP and glutamate, and measure released factors. So in this case, we were looking for only pro-inflammatory molecules because, again, in the PAMP study, we did not find change in other factors. So here, first we measured nitric oxide. Nitrites, LPS was used as a control. You can see that LPS induced or increased nitrite release significantly higher than spinal cord microglia. However, ATP at 1 millimolar concentrations did not affect nitric oxide release. TNF, also similarly, you can see that there was a significant increase for LPS, which was compared to spinal cord was much higher. ATP not have an effect. For IL-1 beta release in media and IL-6 release, we found that, again, similarly, IL-1 beta release by LPS was significantly higher, not so much for ATP. And for IL-6, we did not find any changes for release IL-6 in the media. So first test was for ATP. The second group of DAMs that we tested were glutamate. In this experiment, what we tried is these different concentrations of glutamate were used to mimic different environmental conditions. So 10 micromolar concentration is kind of what is found in normal parenchyma, brain parenchyma. When there is an ischemic injury, that shoots up to 30 micromolar concentration, and anything above that, 100 and above, usually induces excitotoxic injury. So in this case, we are mimicking all these conditions to see if between brain and microglia there is any difference in these pro-inflammatory factors. The first thing we noticed was there was a main effect of treatment. What I do mean by that is between brain and spinal cord microglia, the overall release of nitric oxide was much higher in brain microglia compared to spinal microglia. And for glutamate, however, we don't see any within group differences. Again, with positive control, LPS mediated release was significantly less. TNF was interesting here. We for LPS as well as for glutamate, we don't see any difference in the release of these factors. However, for IL-1 beta, similar to nitric oxide, there was a main effect where brain microglia mediated release was much higher than spinal cord microglia. And similar to our PAM study, we found that within group LPS LPS mediated release of IL-1 beta was significantly less in the spinal cord. For IL-6, though, microglia, we did not find any difference in microglia mediated release of IL-6. So, to summarize the study, we approached the DAMs with the idea that, okay, we saw these differences in LPS or pathogen like mimic. Do we see that? However, we found that when you use 1 millimolar or glutamate concentrations to mimic injury, the overall release of these factors, nitric oxide and IL-1 beta, was relatively reduced in spinal cord microglia compared to brain. And so this suggests that the pro-inflammatory factors, at least for nitric oxide and IL-1 beta, is reduced, and it's similar to that we saw in the LPS study, but not for TNF or IL-6. So, and this is interesting because, and it also makes sense because microglia are very sensitive, and I talked about how the local environment will affect their phenotype. And we see that that phenotype effect could be for particular release factors. It is not for all factors. So in this study, we wanted to see what is the role of environment in dictating microglia phenotype. Is microglia inherently, spinal cord microglia inherently, their inflammation is reduced? Is it inherently reduced? Or is it that the immediate environment is affecting their phenotype? So to test that, what we did is we had brain and spinal cord mixed glia. So when I say mixed glia, they have all the glial cells that would be in a prep, and so this would mimic more like a in vitro brain-like environment. And we had spinal cord mixed glia to mimic the in vitro spinal cord-like environment. And we measured out the media from the mixed glia and it applied it to brain microglia. So what we are actually doing is we are collecting all the soluble factors of the brain mixed glia and we are applying it to brain microglia. And so this potentially would mimic brain environment. And then we took spinal cord mixed glia and then we added that to brain microglia. So potentially mimicking spinal cord microglia environment. And so the hypothesis is if the environment is affecting spinal cord microglia's function, in this case, brain microglia exposed to a spinal cord-like environment should behave like spinal cord microglia. So that's the postulation. And after we did this media change, we measured pro-inflammatory factors, and we picked the two factors that were most prominent in our previous studies, where we found IL-beta changing all the time, and we also looked at IL-6. So again, it's a busy slide. I'll try to go really slow here. We applied all the concentrations that we had, and then the two groups are brain mixed condition media. So basically, brain microglia exposed to brain mixed glia, and spinal mixed glial condition media exposed to brain microglia. So basically, we're taking brain microglia, exposing them to different conditions. And when we measured IL-1 beta released in the media after activating with all these factors, the first thing we saw was there was this minor effect, where when you exposed brain microglia to spinal cord microglia condition, their overall release of IL-1 beta was reduced. And this is very similar to our data from PAMP, where we found spinal cord release of IL-1 beta was significantly reduced. And for IL-6 release, we found that swapping or changing the media to spinal cord like condition had an overall main effect, where there was an increase in overall release of this molecule. Not only that, when we compared across, when we did a Paris comparison, LPS induced a higher release of IL-6, and there was also a higher release by 100 micromolar glutamate. And so this is really interesting because in the PAM study we saw a trend to increase. Here, just mimicking the environment, we have modified brain microglia to behave like spinal cord microglia. So the overall summary is that we identify these changes in pro-inflammatory factors between spinal cord and brain microglia that were changing, and we assumed, or we postulated that this could be an effect of the environment. And so we mimicked the environment, and after mimicking, we found that overall there was reduction in IL-1 beta, and overall there was a reduction in IL-6. So this is telling that the environment does affect the brain microglia function. And not only that, within group we found that IL-6 release was increased, and glutamate mediated activation also increased IL-6. So the take-home message for this would be that the soluble factors in the mixed glia are somehow affecting microglia function. So the differences we see in the spinal cord microglia is a function of the environment. So having said that, all these studies are, PAMP is a mimic of inflammation. ATP and glutamate is a mimic of glutamergic and ATP injury, or ATP mediated injury. We wanted to test something much more relevant to, say, our research, which is stroke. So in this experiment, we designed a model of ischemic injury to see if there are differences between brain and spinal cord microglia. Previously in our lab, Aaron Lai found that when you have cortical neurons that are moderately injured, microglia can rescue them from the injury. However, when they are severely injured, they cannot be rescued. So with that background, we postulated that, okay, now we have our cells, the spinal cord microglia, and they have reduced inflammatory profile, right? So then, if they have a reduced inflammatory profile, exposing them to injured neurons, I assume that it will increase neuroprotection because they're not pro-inflammatory. So that's what we postulated. We said that adding spinal cord microglia to a moderately injured neuron will protect the neuron. And then let's see what the results are. Just a minute. So, if you recall, I said this experiment was to mimic an ischemic injury. So to mimic an ischemic injury, what we did is we cultured cortical neurons and we exposed them to this environment in a chamber where oxygen and glucose was deprived. And then the severity of injury was dependent on function of time. So the longer the cells are exposed to oxygen deprivation, the more severe the injury is. And the shorter they're exposed, less severe injuries. So after exposing these neurons to OGD-like injury, we took them out and then we added brain or spinal cord microglia. So what we have done here is we've injured the neurons and we have applied the microglia at the injury site. So this is like bringing microglia from outside into the injury site and seeing what they do. So after adding microglia, we allowed the system to recover and we measured viability and pro-inflammatory factors. Because the whole idea was spinal cord microglia would protect the injured neurons and to also confirm that this is through the pro-inflammatory factors that was released, we also measured that. So this is kind of to orient how the actual experiment looked. This is neurons labeled in green, active. These are healthy neurons. They have not gone through injury. Nice processes all over. Adding brain microglia just to the control neurons did not affect their phenotype. They pretty much look like regular neurons. Also, adding spinal cord microglia did not affect the phenotype. So this is a good sign to say go, because microglia themselves are not affecting the neurons. How about when we injure them for 30 minutes? So you can already see that with a 30-minute mild injury, the processes of the neurons are almost shrunken, there's more blebbing, and then we add brain microglia to the prep, and we also add spinal microglia prep. There is less of shortening of processes, also for spinal cord microglia. But this is all qualitative, right? When you induce a severe injury, like a 4 hours injury, you can see the cells are really, really unhealthy. Interestingly though, when you put microglia in them, they just eat them all up. There is no more neurons there. Same for spinal cord microglia, they just eat them up. So it's telling that when there is a mild or moderate injury, microglia are trying to keep the cells alive, protecting them. But once that checkpoint is gone, microglia become more inflammatory, they just take up all the cells. So to confirm that, that what we saw, we needed to quantitate this experiment. So for quantitative purposes, we use MTT, which can tell metabolic activity. So and with metabolic activity, we could use that as a proxy for viability. So what we are measuring here is metabolic activity of control neurons. And we set that as 100, and then we applied that all over the brain and spinal cord microglia to set that as 100, and then figure out is there any oscillations in the viability just adding brain and spinal cord microglia. And we found that no, there is not much happening in viability just adding brain or spinal cord microglia. However, when you injure them for 30 minutes, you can see that the viability of controls have dropped from 100%, it has dropped somewhere around 70%. And adding brain microglia increases the viability compared to spinal cord. Basically, spinal cord microglia actually are not protecting neurons here. It's only brain microglia are doing that. And that effect is only in a mild or moderate injury. When you have an injury that's 4 hours long, that protective effect is lost. So this is telling that there is a time window and only neurons introduced to brain microglia are protecting. This was a surprise for us, and we wanted to test, okay, what's happening, because we thought brain and spinal cord microglia would be more neuroprotective, but we found it the other way. So to address that, we measured the same pro-inflammatory factors that we measured prior, nitric oxide, TNF alpha, IL-1 beta, and IL-6. And interestingly, we don't find any difference in these factors. There's no change whatsoever. And if this is representative of the 30-minute injury model. I also have the control and 4-hour injury models. There was nothing happening for any of these factors. So here, the only thing I could say is the brain microglia are protecting these injured neurons by other factors. It's not these release-soluble factors. And they are significantly different than brain microglia, spinal microglia. So the protection offered by brain microglia is significantly higher than spinal cord microglia. So the summary for the OGD injury was, for me it was surprising, because we saw that pro-inflammatory factors were reduced by spinal cord microglia, and so we assumed that they might be helpful or protecting neurons. But on the contrary, we found that there was only brain microglia that protected mild or moderately injured neurons, and not the spinal cord microglia. And I'm also, this is not the first time, right? I mean, Aaron Lai also showed the same thing, where if he has a moderately injured population, adding media from microglia protected those neurons. However, when the injury was longer, when it was severe, that protective effect was gone. So, yeah, it was a bit of a surprise for us. We found that the protection was offered only by brain microglia. So the overall summary of the results is that I started this project with the question: are spinal cord microglia different from brain microglia? And yes, indeed, we found that when you used a pathogen mimic, there were all these marked significant differences in the release of pro-inflammatory factors. However, for physiological activators like ATP or glutamate, that difference was present only in certain factors, not all of them. So nitric oxide and IL-1 beta release was different, but not TNF or IL-6. So that's telling that there is an activator dependent differences in these population of cells. And I also show that, well, even though they start up and they have all these differences, the environment also affects them. So if you have a mimic of a local environment, say spinal cord microglia environment, brain microglia will behave like spinal cord. And so this kind of fits in with all the roles of microglia. They are very sensitive to pathogens. They are also sensitive to physiological activators, but not all the effector molecules are different, and the environment plays an important role. Changing the environment will affect their function. And finally, we found that brain microglia are protective compared to spinal cord microglia. And for now, we can only speculate as to that there are other factors in the media that brain and spinal cord microglia were introduced and released. That's not the pro-inflammatory factors we measure. Right? So that, and what would be the overall conclusion of this thesis? So that's where I'm heading with this. First, I started my research in 2011. I said, well, these are cells that come from a common progenitor. Are they different? And we tested that, and we found, yes, they are different. And this is not surprising, because not only us, in the past 5 to 6 years, many labs have found that there are regional differences in brain microglia. And so what does it mean for us is that there is no one-shot cure for inflammation, or one-shot solution to cover the entire CNS. I mean, we need to find better ways to regulate inflammation, because we know that these cells respond differently from their region of origin. The other thing is, which is expanding on the thought, is that brain microglial response to pathogen could be different compared to spinal cord microglia. There is somehow where spinal microglia are attenuated or reduced to their response when they're exposed to pathogen, which is interesting. Even though they are on the same CNS, brain microglia are more responsive to pathogen-like stimuli, and spinal cord microglia are not. The third point is, so HAMs and DAMs also induce a differential response. So with pathogen, we found it was like a yes or no. All factors change and did not change. Versus when you use more like a physiologically relevant marker, activator, we found only some factors change, and that change was not present in all conditions. So the final, not the final, penultimate is that it's not that only that there are differences based on the activators, there's also differences based on the environment. Swapping microglial environment can affect your function. That's telling that these cells are also very highly plastic and the phenotype is not fixed. And this could have potential for treatments where we could modify microglia to a more anti-inflammatory like state, and maybe if we introduce them into the site of injury, they might help with the recovery of the injury. So that's the first 5 important points, four points that I want to stress. Final point is that all these differences that I saw actually had a meaningful difference in their neuroprotective role. So when you introduce brain or spinal cord microglia to an actual mimic of an ischemic injury, it's only in the brain microglia that we see a difference, where they were more neuroprotective and spinal cord microglia was not. And that leaves me with this question as to, okay, what is happening? I have really no idea. Is it the soluble factors that are released? Is it inflammatory factors? I don't know, because we tested pro-inflammatory factors did not change. So that's an open-ended question I'm trying to still answer. I think with that I'll conclude my presentation. I really like to acknowledge my entire team. They've been there for me all the time, especially Matt. Matt's been my third mentor. He's almost like a supervisor. Every time I go knock his door, he has answers for me. Appreciate the support from Patricia and Gail. Gail has been great support for my my entire PhD program. I also thank the examiners for having the time to come and listen to my talk. I also thank Chairperson Monica. Thank you guys. It's really good to have you all. Especially I can't forget my lab mates. The whole crew is there. Thank you guys, and my friends. I have friends from all over the city. They're all like there. So it's good to see you guys. Thanks for coming. Finally, glial cells and their love. No, this is not Photoshop. This is an actual mixed glial picture that I took from my prep. They send their love. Questions, please. Thank you.
Thank you, Sam. All right, does anybody have questions? Oh, a reminder for those of us on the examining committee, we do not ask questions at this time. Everybody else, please.
Right. Sam, that was great. Thanks. So you showed that spinal cord microglia seem to release less inflammatory cytokines relative to brain microglia. Are there more microglia in the brain relative to the spinal cord? Okay.
In percentage-wise?
Yeah. Like, do you find just greater densities?
So there are studies that show that within certain regions of brain there are higher densities, but the overall density is around 5 to 15% in the brain, and in the whole of spinal cord it's also around 5 to 15%.
Okay, so when you did your isolations, you got about the same number.
Yeah.
Okay.
And the seeding densities are always kept the same, because that is a discrepancy. When you have higher density of microglia, you're effectively mimicking an injury state. Yeah.
So from my understanding with this study, basically the spinal cord and the brain have to go reactively to the stressor, right?
Yeah.
You're going to be studying MS when you get in the drug. Does that mean that for any sort of drugs that you need to engineer, they will reactively to different areas that they're being used to treat?
Yes. No. Absolutely. And that's why we really want to test these things, because some of the drugs are effective in culture, but when you apply it on an actual animal, it doesn't work. So it's not just one factor. There are multiple factors that would confound why a drug would not work, but the heterogeneity possibly plays a huge role as to why we have these discrepancies.
It's really important to
Mm-hmm.
And my question for you then is, well, a couple of questions, I guess. first one is, in general, is there a difference between gray matter and white matter microglia?
Yeah.
A lot.
Yeah.
Big difference.
Yeah. Well, I wouldn't say big difference, but there are morphological differences, of course. The morphology is different. At least within the spinal cord injury, they found different subsets of microglia behaving differently. So I'm speculating, yes, there are differences.
So here's the second one. Is there a different ratio of gray to white matter between spinal cord and
The brain.
so
Yeah.
that you're not necessarily dealing with differences in brain spinal cord,
Yeah.
but just in the ratios of the gray white matter?
Exactly. So that's why I was more kind of globally saying environment plays a huge effect in how these cells respond. So, it depends on the environment. And it's more so about the local environment rather than, like a global state. Microenvironment.
And to follow up on that,
Yeah.
Phil Popovich has actually shown us a while ago that a different level of spinal cord inflammation has a different quality. So I guess when you raise your cells, all the microglia, you might actually look at the different levels and there might be a difference.
Yeah. So right now it's technicality. I mean, it took me some time to actually get this to work. And so, looking at within populations, it's hard because the yield is always low. Brain seems to have an abundance of microglia versus when you try to isolate from the spinal cord, there's always a lower yield. So, sorry.
Completely different because I actually forgot how it was done, but it was the good old word from Couch Starks who kind of activated macrophages. Kind of similar idea that were then used to treat, never really made it to the clinic, but really in clinical trials to, as a neuroprotective and cholestatic treatment. So that's kind of a similar idea. How did she activate them? Do you know that? And would you have to maybe activate the microglia in a similar way to actually see any protective effect?
I don't know that study, so I can't really tell. But so activating them, it's interesting, right? I mean, in stroke, there's one study I can actually relate to that. When you have microglia introduced immediately after stroke injury, it actually benefits, it reduces in fat volume. Sometimes the very presence of microglia facilitates recovery. And deletion of microglia increases, exacerbates injury. So even if we activate them, how do we know that for sure they're going to maintain that status? Because now they've gone to this environment where they respond to that environment. So that's something that always, I'm always pondering. Does it really matter to activate them before we inject them? But priming does. I think you have shown that when you prime them with players, so. Yeah.
So one of your initial slides, you were saying that under basal conditions, neurons will release factors that keep microglia amplified. So is it known what those factors are? And then also, is it once the neurons are injured, are they releasing more of those factors to shift them to a mucoid, or are they stopping?
That's a very good question. Very good question. You have to remind me the second question because I'm trying to work out the first question. Okay. So there are a couple of factors. Some of them are membrane bound, some of them are released. The major ones that can come to my head right now are CD200, fractalkylin, TREM2. These are all factors that will tell, hey, I'm here living, don't eat me, kind of cells. So yes, these are there. But when you, for example, for fractalkylin itself, it's a contact receptor. Right? When microglia lose that contact, they think, okay, I lost a neuron, I should probably eat it, and so it attacks it. And so yes, there are. But so like I said, you have to remind me your second question.
Yeah, the second question was just if those factors that are released, once the neuron's injured, do they stop releasing them, or do they release more, and is that what shifts?
So when they're injured, they're dynamic to release. There's a tendency to release other factors. Like if there's a metabolic deficit, they would dump ATP and glutamate into the system. So there will be a shift in the factors that they release, and that's why I think with a moderate injury, you have a mixed population where some of them are healthy, some of them are not healthy, and so somehow microglia are able to recover these cells. versus when you have complete injury where all of them are, completely injured. There's no room to restore these survival signals. So, yeah.
Is there any reason to think there might be a fundamental difference in the susceptibility of the brain spinal cord to getting an infection or integrity?
I think so. The only problem is there was a study where they used a virus to infect spinal cord, and they found all the factors that they showed were actually upregulated in the spinal cord. And so, one of the reviews is saying, no, they show that an upregulation what is happening. So, I think if I have to speculate, what I'm going to say is, at least with the spinal cord, the tendency for blood-brain barrier to be leaky is much more higher than brain. So maybe microglia interact with peripheral immune system more in the spinal cord versus in the central nervous system, which means now they have support system that they can immediately recruit. So maybe they don't have to do the same amount of work that a central nervous system, brain microglia is doing. But that all goes into the world of speculation.
All right, no more questions? Thank you very much, Sam.
Wait. Once again, thank you very much. Oh, thanks.
Yeah, we go up to CSV now.
Yeah, I'm packing up and I'm coming.
That's just what you needed to do.
Thanks.
That's very good.
Was I too quick, too slow?
You were calm, you presented it well. I thought that was perfect.
Yeah, I had my moments where I was a bit struggling, but yeah.
It was clear, you took your time, explained everything, and walked us through it. Congratulations, buddy.
Thanks, man. Yeah. Thanks,
Facebook.
Sure.
Well done.
Really?
That was awesome. Yeah.
Thanks, man.
That was
Yeah.
so good.
வணக்கம் தலைவா வந்துட்டீங்க போட்டோ எடுத்துருவோமா எடுங்க உங்களுக்கு என்ன
நிறைய எடுத்துட்டேன் அனுப்புறேன் அப்புறம்.
ஆமா அண்ணா சிவானா அருள் அண்ணா ரெண்டு பேரும் Yes, boss.
Damage is so
Yeah.
danger up again.
Yeah. Thanks for cracking. I think
What's that? No. You had it right on one side. But yeah, you got a couple minutes.
yeah. I have to go to my final defense, guys.
Yeah.
Yeah. But yeah.
Good luck.